Hello community! Does AI generate economic value for our society? Today we look at new drug discovery processes with AI. So we are deep into bioinformatics, and the question is simply, hey, how can we use AI and what we know about AI? And especially we will deploy multi-AI agent systems for bioinformatics. Now, our input is simple. We have here a chemotherapeutic drug name and a specific, let's say, a human target protein. And we just want to know, will those biomolecular structures bind together? Will it work maybe as an inhibitor? So let's say the simplest example here, topotecan for the drug and top1 here for the target protein. Now, what is happening? We have now an AI agent that retrieves, let's say from a database or wherever, here a specific string. This is called here in bioinformatics the SMILE string, a simplified molecular input line entry system for the drug, which is nothing else than a textual representation of the precise molecular structure of our drug. And of the other side, of course, we need for the target protein, we also need here a three-dimensional, if you want, biochemical compounds information, and it fetches here the amino acid sequence of our top one target protein. Therefore, we have now quite some information, so let me show you where we can find all this information and how we will use it. So we go NIH, National Library of Medicine, we put here our name, here we have it, we just click on it and we have here some basic information on topotecan. As you can see, if we look here at a three-dimensional, let's animate it a little bit. So here you see, you have an idea about the three-dimensional structure of our biomedical compound. And now, of course, we have a lot of information, as you can see here on the right-hand side, about toxicity, about all the patterns, about taxonomy, about classification, all the name, all the identifiers. Here we have our canonical SMILEs. This is exactly what we are looking for, isomeric SMILEs if you want. So here is all the information available, of course, also for our target, the top one inhibitor. Here's our three-dimensional structure. It has a beautiful name if you want to pronounce it. This is, of course, great. And also we have here all the information that we need. Beautiful. So let's build our first agent. I will give you the code. It is open source, the code, but I just want here to focus here that we really are on the same level, that we absolutely understand what we're going to do with multiple agents and how they will interact. Agent number one is a simple one. We just go to a Python library and we do here a computation. So we will use, of course, here some Python, PyTorch, libraries. We have DeepPurpose, so it takes an input here, the SMILEs of a compound and a protein amino acid sequence, and then it generates here, simple as you know, embeddings, vector embeddings. So here we have our first step, our compound. Here you have here the SMILE code if you want. And then you go either with a CNN or with a transformer, and you generate the compound embedding of this particular compound. Great. For the protein, we have also a protein encoder. We have a very specific protein. We have the amino acid sequence of this protein, and we do exactly the same. We have here a neural network. Choose a transformer or whatever you like. I will show you in a minute a specific combination of a graph neural network here. And what we do, we create protein embedding information on a high-dimensional vector space. And then if we have the compound embedding information from the compound encoder, and we have the protein embedding information from the protein encoder, we have now a decoder in our transformer network. You're not going to believe it. And the output is simply a value that tells us, hey, will those two biochemical compounds bind together? Will we have a chemical reaction or not? And this is simply, if you want, a computer simulation based on all the empirical data that we have. And we will train here a neural network, a transformer architecture to predict this for new compounds. This is more or less the agent number one that we're going to build. Now, of course, we have empirical data. Society has a binding database here, where they specify here exactly for a kind of a neural network that is a combination of a message passing neural network and a convolutional neural network. And we have here the training data here for our system from the binding database. So we have an architecture, we have the training data, and let's just train here this neural network architecture, this machine learning model architecture for the prediction of the drug-target interactions. Great. Now for my green grasshoppers, very short summary, message passing neural information. Why we do this? It is designed to work with a graph-structured data format, which is ideal for modeling the molecules. So in the context here of our drug discovery, each molecule can be represented as a graph structure, simply where the atoms are the nodes and the bonds are the edges. So what we do, the message passing, the information is passed between the nodes along the edges, and then the network can capture here the local structure and the connectivity. After the message passing, the node representation updated based on the information they received from the neighbors. And finally, we aggregate here the node level features to generate a whole graph representation, which then can be used to predict properties like the binding affinity. Great. Now, we will use a combination of both of this here with a convolutional neural network. Now, my green grasshoppers, convolutional neural networks, we use this in the old times for images, so two-dimensional somethings. Why? Because they are really good here to process the spatial and the structural features of the molecular binding sites. So we have here the classical layer, the pooling, the fully connected layer from our classical convolutional neural network architecture from, 5 years ago. And then what we do, we combine it here. Why? Because here the message passing component processes here the molecular graph of the drug, capturing here the structural information about this particular molecule, and the CNN component processes here the spatial features of the binding site of the target protein. So you see, for both substances we have different compounds here or different architectures, but if we combine them and we run them through a specific neural network, this can now predict here the binding affinity between the drug and our target. So great, we have now our architecture. Again, as I told you, we have here the binding database data available from thousands and thousands of substances and proteins and genes and whatever we have. So we have here our database, our pre-training data set that we're going to use. And then we have here, once trained, a beautiful neural network structure, maybe a transformer network, to predict the binding potential of new or repurposed drugs against new targets. I will give you the code in about 2 minutes. It is open source code. You can take it, you can play around, you can experience this. But I told you we will do a multi-agent. So this is just one agent who does, let me tell you this in a very simplified way, the scientific calculation on a molecular level based on empirical data of thousands and thousands of proteins and genes and compounds, where we train a neural network on a prediction, but we want to have a verification. So we need another agent. Maybe we have construct 2 other agents. Now, of course, we need here tools. And as I told you here, we go with December 2020. Harvard University published this together with Georgia Institute of Technology and IQVIA, Cambridge, and University of Illinois, DeepPurpose. DeepPurpose, if you're not familiar, is a deep learning library for drug-target interaction prediction. Real beautiful paper. Of course, they have here the GitHub repo that you can go to, deep learning toolkit. And you have here everything from protein modeling to protein function prediction. It's a Jupyter notebook in Python. Beautiful, from 2020. So code for agent 1, clear. Agent 2 is what you know. We build now a knowledge graph agent because it is simple. We have so many databases. We have so many biomedical databases. Let's focus on human, for examples. We have, or whatever you want to go with, we have a drug bank. We have a comparative toxicogenomics database. We have a drug-gene interaction database. And we have here prediction about chemical protein interaction stage. So all of those databases, the knowledge of those databases, now we write a short program so that we can now construct a knowledge graph based on the information in 1, 2, 3, 4 databases. And then what we're going to do is simply on the knowledge graph itself, we're going to calculate DTI. And you might ask, how is this possible? We do not have a biochemical or physical computer simulation behind. No, we will use here something real simple. If we have a knowledge graph, we just calculate here the shortest path between the drug and the target protein within the graph. And the number of hops that you know from really old time ago. So the number of connection from the drug node to the target node is now a kind of an indicator for us how closely related those are. Because if we have two biochemical substances, compounds here in a database, and there has been a lot of research on the combination of those two compounds, then they are, if you want, closer together. So we have a direct link and we do not have some connection over another connection over another connection, and finally we come to the target node. So you see, this gives us kind of an indication how close the interaction of those biomedical compounds are already researched on, now are available in different databases. If you have other databases, please go with your database. This is just here an example. And then, of course, we build a third agent. Yes, we want another validation here. And what is the simplest you can think of? We just have here a search agent. Maybe you go for an internet search, you connect to a proprietary database of your university, of your, I don't know, pharma lab, of some companies where your university pays that you as a student or whatever you are, you get access to those data. Whatever you have, or you just can go to PubMed, or you just go to Google Scholar. You can go to any source of scientific papers that your university is paying for, and there we simply go and we formulate here a query combining here the drug and the target name, and then we, in the simplest case, we search here for a combination or interaction here in those databases. Or if we have some other publication, you know, this is where we have this web scraping. So we search here the web, that are all the sources that allow us to search those sources. And then we get maybe new titles, new abstracts, or new information on this specific drug target interaction that were just published, I don't know, yesterday or just some days ago, and are not yet available in all the classical databases that we have. And what we do, how do we calculate here the closeness or the established interaction between two chemical compounds on a molecular level? We do a text analysis. It couldn't be easier. You see how easy it can be to build an agent. We just say, okay, so we have now a keyword detection about interaction, strength, what about binding, inhibits, activates. We look for all the possible combination of those words, and we just analyze how are they now in the text, in the semantic information provided to us, combined. And this is it. We have 1, 2, 3 agent. So here we are, bioinformatics. We have our biomed agents. We have a multi-agent system. And what we do, we simply leverage state-of-the-art dedicated biochemicals LLMs, knowledge integration from various biomedical databases, and an automatic literature review to predict drug target interactions. And please note, this you can do even if you're not in bioinformatics, if you are in material science or in theoretical physics, or you are I don't know where, and you have to find combination of different materials and you want to know more if the molecular structures are similar, if they would bound, what would be the next chemical reaction. There are so many databases available, and here 3 simple agents, and let's have a look how they are. We know now there is a Python library available for us that does all of this. We just need to input here the name of the compound and our specific protein that we are interested in. And I would like to show you that those research teams here, University of Minnesota, and Rensselaer Polytechnic Institute, they did this. And they created this drug agent, as they call it, explainable drug repurposing agent with LLM-based reasoning. Because if we get it back here from the text, of course, we have an LLM that understands here the text that comes back from the databases. And there are some causal reasoning structure. Yes, we can enhance them. You can enhance them with your code that you have. But the nice thing is that they made available their code here that they used here. And this is beautiful. And this is the reason why I selected this paper to show you today. So we went now about the theory. We built theoretically 3 agents. And now for the code, it is rather easy. Let me show you the process maybe before. So we have a user input, and you have designed 2 specific questions about a drug and a target, as I just showed you. Then we have here a fraud agent. I have not mentioned this yet. This is here our super agent, our coordination agent that distributes the work to all our 3 other agents. You can imagine, yes, beautiful. Then all those sub-problems are solved now by our machine learning agent, by our search agent, and by our knowledge graph agent. They have now their reasoning process integrating now a multi-source information coming back. So from the machine learning prediction, we have now a specific value. Let's say this is 7.65. From the literature, we see that nobody discovered this. We have 0.2. From backward information about something else, we get some additional information and we can come to a conclusion. You might even say, okay, for me, it is really important to focus here on the mathematical computer simulation. So you weight this with, I don't know, 70%, and the other 2 agents, you just distribute the 30% left. You choose the weight whatever you like, but you have 3 active agents that generate results on a different level of complexity for you. Great. So if we apply this now, and this is the nice thing here, the prediction can direct laboratory experiments to validate potential drug repurposing candidates. So we already have some potential medical drugs that could help us in other cases. And now we don't just want to do here the human experimentation, but we just do computer simulation. And AI should tell us, hey, is this potential drug that we already have, can we use it also for other illnesses to treat those things? Then, therefore, we can identify new therapeutic use, and it will support the clinical decision making. Prediction can assist in selecting specific drugs, here maybe off-label use, based on their potential against specific targets. And we are talking here about your specific genome structure, your specific medical conditions, and you see exactly we are going here in the area of individual medicine. Future direction is very nicely because we can autonomously update our knowledge. We can let the AI system simply run and discover new patterns, discover new potential candidates. We can handle now complex drug combination because maybe we just have not only one drug, but maybe we have a combined effect of 1, 2, 3 different medical compounds. We have maybe better insight into multi-drug therapies based on a computer simulation of the biomolecular compounds, if they're gonna bind at a molecular level, yes or not. We can improve our scientific models, and we can improve, if the models are evaluated, then also our therapy. And as I told you, we can have then more specific, more targeted treatment to have here, or to make here even personalized prediction by our multi-AI agent application in the future. So there we have it. DrugAgent, really nice study published about 2 weeks ago, and the code on GitHub. Let me show you the code. I give you the link to the GitHub repo in the description of this video, but you have here, you see all the agents. There you have your integrator agent, you have your config JSON, you have your integrated agent, the knowledge graph, the probability calculator, the search utilities. Here I go with the machine learning utilities for our first agent if you want. You utilize here, as I showed you, the library DeepPurpose. We import here our data set, we import the utilities that we need, and we import, of course, the model that we're gonna need. And you have the code here available for you. As I showed you, we use here this particular model that is already available for us. This is here this message passing neural network here with this convolutional neural network, where the data that the system architecture has been trained on, where the binding database data here from thousands and thousands of our candidates, and you simply have here the code. If you want anything, please have a look. You have here data even where you have all the training data. If you do not have access to the online libraries, but normally your university should provide you a link to all this biotechnical information, then we have the pre-process. Yes, beautiful. Yeah, and in the end, if you implemented all of this, you just have here a very simple script. So for the first agent, for our machine learning, we have here the specific smile string for our specific drug, if you want. And then we have our target encoded here. A sequence is smiles. Then for our knowledge graph utility, the Python file, you have this one. And then for the search utility, it couldn't be easier to use here those scripts. Those are available. You see, last update was August 21st, 2024. And if you have stayed with me till the end, I want to give you a short bonus. If you want to continue in this direction of research, I would recommend this particular paper for you. It is called Genesis Towards the Automation of Systems Biology Research. This is by Chalmers University of Technology, Sweden, Royal Institute of Technology, Stockholm, University of Cambridge, Vanderbilt University, Nashville, University of Manchester, UK, and University of London. Beautiful. End of August 2024. This is now about robot scientists performing here the experiments. So this is really the next generation robot scientist for system biology experimentation. Really interesting if you are into these topics, and if you want to add to your knowledge here also the robotic component, because I guess that our research labs will change significantly in 5 to 10 years. This is the paper you might enjoy to have a specific look at. I hope you enjoyed it. I hope there was some new information for you, and it would be great to see you in my next video.